Hello everyone, welcome back to biology class. Today in this video lecture we will study about embryonic development in human. Student, in last video lecture we have studied entire process of fertilization where male and the female gamete they fuse together and it forms the zygote. The diploid zygote, which is produced in process of fertilization, it undergo mitotic division, that is the cleavage. It produces morula, blastocyst, and the gastrula. These are all the stages we are to study in detail in this video lecture. Cleavage, it is the early embryonic cell division. It is taking place in the zygote. During this cell division, daughter cells are produced called blastomeres. The size of the daughter cell get reduced in every successive cleavage. In daughter cells or in the blastomeres, the metabolic rate is very high, as well as rapid replication of DNA and the high rate of consumption of the oxygen, it is also seen. And that's why the cleavage rate is the fast. Look at this animated picture. We can see how the cleavage it is undergoing in the zygote. Let us see how this cleavage process it is completed in the zygote. The cleavage it is a holoblastic, means entire zygote it is divided by the cleavage planes. There are two planes in the cleavage, longitudinal and the equatorial. Longitudinal plane in which zygote it divide vertically, and in equatorial plane zygote it divide horizontally. The first cleavage in early embryonic development it is a longitudinal. After the 30 hours of fertilization it begins. Cell produced are termed as blastomere. In first cleavage, two blastomeres are produced. One, it is slightly larger than the other one. Look at the figure, how this longitudinal division, it divides the zygote into two blastomeres. This stage, it is called two-cell stage. The second cleavage begins immediately after the 30 hours of the first cleavage. This cleavage plane, it is also longitudinal, but it is a right angle to the first cleavage plane. That means the first cleavage plane, it is also longitudinal. Second cleavage plane, it is also longitudinal, but both the planes are right angle to each other. Like we cut the apple. The first cut is also vertical. Second cut of the apple we take, it is also vertical, and it gives the four equal size the pieces of the apple. In the same way, here the zygote it also is cutting in the second cleavage into four cells. Here the cells which is produced, they are the blastomeres. The stage it is called four cell stage. Third cleavage plane, it is horizontal. It cut the zygote horizontally. The four cell structure, now it is converted into 8 cell structure. The embryo, now it is pushed towards the uterus. In fourth cleavage, 16 blastomeres are produced. 16 cell structure, further it divide and it gives 32 cell embryo structure. It is looking like mulberry fruit. This stage it is called morula. This stage it comes after the 4th day of fertilization. It has two types of the cells. Smaller cells they are present at outer side, and the larger cells they are present at inner side. The morula cells they are arranged very compactly. They don't have any space in between. and Zona pellucida, it is intact around the cell. It is retained till the embryo it is get implanted. The size of the zygote and the size of the morula, it is the same. Only the number of cells it is increased, but the size it remains the same. At the end of the fourth day, morula it enters into uterus. Morula it is pushed into uterus and it remains floating in the uterus. The outer cell layer of the morula cell, it forms a trophoblast layer. Trophoblast cells, now they start absorbing uterine milk. It is a nutritive which is rich in glycogen. Now this trophoblast cell, after the absorption of uterine milk, they becomes flat and it helps in the absorption of further nutrient for the growth of embryo. Inside the trophoblast cells is the inner mass cells. These are the actual embryo cells. They are going to develop embryo. By the end of the seventh day of fertilization, the trophoblast cell, it forms the contact with the embryonic cell. It is called cells of Robber. At this stage, embryonic polarities also form. The inner mass cell, which is in contact with the trophoblast cell, they are called as the embryonal end, and opposite to that inner mass cell, the end it is there, it is the embryonic end. Look at the figure. In the animated picture, we can see inside the trophoblast, here the cell mass it is collected at one side. That end it is the embryonal end, and the end opposite to embryonic mass, there is no cells, only the trophoblast layer is present. That end it is the embryonic end. So that indicates the embryonic polarity. Student, look at the picture. Here we can see early blastocyst present in the uterus. Early blastocyst has outermost zona pellucida, it is still intact. Inner to that, trophoblast cells. Trophoblast cell, it covers the inner mass cells. As the inner mass cells, they are gathered at a pole. The rest part of the blastocyst is having the cavity. It is called blastocele. The formation of this cavity, it is the characteristic feature of the blastocyst. It is formed after the seventh day of fertilization. Till the formation of the blastocyst, zona pellucida, it is intact. It prevents the implantation of embryo at abnormal site. When embryo get a proper site, then only zona pellucida it will start disappearing. That is after the implantation. With the help of this animated picture, we can memorize the changes from the zygote to the blastocyst formation. After the day 7th, the implantation process it start. Implantation, it is a stage of pregnancy. At this stage, embryo it attach to the wall of the uterus and start receiving oxygen and the nutrients from the mother's womb for the growth. Student, look at the picture. Here we can see the trophoblast, which is the outermost cell layer of the blastocyst. It is attached with the endometrium. Endometrium, it is the innermost lining of uterus. This attachment, it is seen only after the 7th day of fertilization. Student, now let us focus on the implantation process. It is similar to the sowing of the seed into the soil. Here, the seed is blastocyst and the soil is the endometrium. The seed, it is sown into the endometrium. The blastocyst, it is having the trophoblast cells. These trophoblast cells, they are attached from the inner side to the inner mast cell. The point of attachment of inner mast cell with the trophoblast, this point of attachment, from that point, this blastocyst, it start implanting into the endometrium. See the figure. You can find here the blue color structure, it is the inner mast cell, and the trophoblast cells now they start dividing, and it gives the two types of the cells, syncytiotrophoblast and the cytotrophoblast cells. Syncytiotrophoblast cells, they are present in front of these inner mast cells. The cell boundaries of these cells dissolve. Only the outer cell boundaries, it is present, and that's why we can appear this cell structure in the form of the finger-like projections. These finger-like projections, they enter into the endometrium to derive the nutrition for the further growth of embryo. The rest cells of trophoblast, it is a cytotrophoblast. This stage, it is appears on the 10th day of the fertilization. Since cytotrophoblast, they have the lytic enzymes. These cells, they release their lytic enzymes in the endometrium cells, and because of that, endometrial cells, they get ruptured, get digested, and slowly this blastocyst, it get implanted. Implanted blastocyst, it is come in contact with the blood vessels of the endometrium cells and the blood sinusoids of the endometrium and start deriving the nutrition. So in this picture, we can see here the growth of this endometrium. Endometrium, it becomes the thick. It becomes the thick and it is waiting for the implantation to maintain the pregnancy. After the implantation, the next stage of the embryonic development, it's begin. It is the gastrulation. In gastrulation phase, the rate of the cleavage, it is slowed down. In this process, the formation of the gastrula, it is done from the blastocyst. During the gastrulation, the cell movement, it result in a massive reorganization of the embryo from the simple spherical ball of the cell, that is the blastula, into multi-layered organism. Two important events it is taking place during the gastrulation, and these are differentiation of the blastomeres and the morphogenic movements. So let us see what are these movements. In gastrulation, the movement of the cell, it is seen. Because of this movement, the bilaminar structure, it is formed in the inner mast cell. So look at the figure, you can see here the bilaminar structure, as well as we can see over here syncytotrophoblast, which is formed the finger-like outgrowth, where the multinucleated structure is present, and this structure is also having what? Lytic enzymes. So you can find in this figure the bilaminar structure and the syncytotrophoblast structure. In bilaminar structure, The upper cell layer, it is epiblast, and the lower cell layer, it is a hypoblast. You know, these two layers, it is differentiated by the inner mass cells. The epiblast cells, they are in close in contact with the trophoblast cells. These cells, now they are going to develop into the ectoderm cells. And the hypoblast cells, see the figure, the hypoblast cells, they are going to develop into endoderm structure. So these are the three pictures which is given to you. It is of what? 7, 8, 9th day of the fertilization process. Now we'll study how the ectoderm formation it is done in the embryo. The epiblast layer, it redivide, and it gives the cells. They get arranged and it forms the cavity. This cavity, it is called amniotic cavity. Look at the figure. In that we can see how these epiblast cells, they grow and they surround and it forms a cavity which is just present above the epiblast cells. The cavity which is formed, it is called as the amniotic cavity. This cavity cells which are in close contact with the trophoblast cells, later they produce extra embryonic membranes. The floor of this amniotic cavity, the pyramidal cells which are present, these pyramidal cells, it is going to develop ectoderm, which is a germ layer. Hypoblast cells, they also start dividing. They divide, redivide, and it encloses the part of blastocell. You know the blastocell, it is the cavity of the blastocyst. Now in this cavity, hypoblast cells, they divide, redivide, and it encloses a cavity. This cavity, it is called yolk sac. So here the two cavities are produced. One is the amniotic cavity and the lower one, it is the yolk cavity or the yolk sac. Student, up till now we have studied about the formation of the endoderm, which is the first germ layer which is produced in the embryo from the hypoblast cells, which enclose the yolk sac. Same way we have studied about the epiblast cells. The cells, they divide and it gives amniotic cavity. The roof of the amniotic cavity, it gives further the embryonic coverings, that is amnion and the chorion later on. The floor of the amniotic cavity, where the columnar cell or the pyramidal cells are present, these cells, they are differentiate into the ectoderm. So up till now we have studied the bilamellar embryonic disc formation and the formation of ectoderm and the endoderm. Now we'll see the next germ layer, that is the mesoderm formation. In mesoderm formation, the ectodermal cells present at the floor of amniotic cavity, they start moving inside the hypoblast cells. They create the groove. This groove, it is called primitive groove, which later it develops into primitive streak. The primitive groove, where these ectodermal cells or the epiblast cells enter, they divide, redivide, and it gives the layer between the ectoderm and the endoderm. It is a layer mesoderm. So with the help of this picture, we can understand this formation of mesoderm. So how this epiblast cells or the ectodermal cells, they are moving inside. It is forming a groove. This groove, it is called as a primitive groove. This primitive groove in which these ectodermal cells are present. Now this groove, it is growing inside, and from the lateral side, the cell growth which is present, this lateral side cell growth, it gets spread between the ectoderm and the endoderm, and it gives the third layer, that is the mesoderm. Now we'll study how the primitive streak it is a form. We know the epiblast cells, they start growing inside. They migrate inside. They form the folding structure, and this folding structure it gives the groove. This groove it is called primitive groove, and the cells which is present in the groove it is the primitive groove cells. This groove slowly it is increasing its length. As the cells they are added in this groove, the cell here the groove it increases its length and it appears like a streak. The base of the streak it is the posterior end of the embryo, and the anterior growing point of the streak it is the anterior end of the embryo. So in the figure you can see how this streak it is growing. This streak it reached to the yolk sac. In the third picture you can see the streak it is reached to the yolk sac. It communicates with the yolk sac by an opening it is called blastopore, and this blastopore it will be the future anus structure in the human being. Student, we have studied the blastomeres. They are differentiated and they are given the three germ layers, that is the ectoderm, mesoderm and the ectoderm. These are the primary germ layers. They are formed as well as they are organized in their proper location during the process of gastrulation. After the process of gastrulation, the next process is the organogenesis. In the organogenesis, the primary germ layers, they are going to develop into the different internal organs. Look at this chart and see which are the different organs it is developed from the ectoderm, mesoderm and the ectoderm. Hope you understood today's topic. Thanks for watching. Please subscribe for more videos.